Terrible.
I'm planning to go back and watch it.
I'm sorry.
We'll go over this.
The things that I expect you to know are less than what I'm putting up here.
Oh,
Less than what I'm putting up here.
Okay.
So,
Don't write anything else.
no, no, you still need to write it all. Okay. Oh. So up until this point, most of the energy from the sugar, most of that sugar's energy is still in these. These are carrying a lot of the potential energy that came out of that sugar molecule. So the whole point of electron transport chain is to convert electron energy into ATP.
And then that electron energy, that's coming from the NADH and FADH, right?
Yes.
Okay.
And your book does not act like FADH2 exists in the pictures, but it does.
Wow, they just
And we
need
need
out
to know
important
that.
information.
Well, I mean, you've heard this stuff before, right? Yeah. No?
I might have heard it.
But was it
My freshman
retained?
year of high school.
And they don't talk about this?
He had us draw pictures of plants.
Okay.
It wasn't very good.
It really good in the 18.
Okay, good.
I haven't gotten there yet.
Yeah.
I don't know.
So when you do, this is like the microscope. When you do get to it in AMP1, it's a lot of this same stuff again. And he probably, I like it when he and I have really different teaching ways. And I like for students to have different teaching ways because it helps. Maybe he explains it better than I do, or different strokes for different folks, but. So, I just thought, does anybody even know that show?
My dad watches it.
I actually never watched it. I didn't watch much TV when I was that age. Long time ago. But the whole point of any electron transport chain is to take energized electrons and turn them into ATP power. So for this class, there are two styles of electron transport chains.
My God, I ain't got enough paper for this.
What?
I'm using it all over.
I'm sorry, Brooke. What about in that binder? You got more paper in your binder?
No.
No.
Just write it up here in this one.
You
So draw a pen.
Okay. There's two styles possible for electron transport chains. You can write this over here. You can write it over here.
I thought it was just,
Aerobic. This can be aerobic or this can be anaerobic.
Oh, I was stressing about that.
Okay. Write it right here. Aerobic or anaerobic. And the difference in those is what is the final electron acceptor? They work basically the same, except what's different is who at the end of the line, circle around these. Draw a circle here. And make a note in this space right here, final electron acceptors.
That little group right there?
Yeah, so it's oxygen, nitrate, that one's nitrate, which you don't have to know.
Oh, good.
You know that though.
In the
You've had
book.
chemistry. Sulfate,
I gotta take that class.
sulfate. So these are the possibilities of final electron acceptors. And if oxygen is the final electron acceptor, that is an aerobic electron transport chain. Like aerobic goes with oxygen. So everything that you learn in AMP1 about electron transport chain and everything that you learn in human biology about how we burn sugar All of that's aerobic. So every metabolism style you learn about before this class, it's aerobic because it's using oxygen at the end of the line.
What makes the cell decide to release the oxygen instead of like the nitrate or the sulfate?
Okay, so what makes it decide to use?
Yeah,
Release, but it's use. It's a spending, using it up. I love that question.
exact same steps.
Yeah, but they are, whatever these electron receptors are, they're picking up the electrons and then they're turning into something else. So what would be released is water. What would be released is nitrite. What would be released is sulfite. Yeah. So the way that they make that decision, that's a big deal. And I'm going to answer that question after I write this next thing. So if oxygen is the final electron acceptor, that's an aerobic electron transport chain. And if it's any other chemical, it's an anaerobic electron transport chain.
And then what did you say to put around that section right there? Electron receptors?
These are final electron acceptors. Like this, final electron acceptor. So the decision on whether to use oxygen to take those electrons or whether to use something else depends on this question, Hawken. Are both electron transport chain styles available? So for people, we don't have an anaerobic option for this. All of our electron transport chain equipment is set up to use oxygen. But there's some microbes that could do either. So when we talked about facultative anaerobe, they have both styles.
But then like an obligate aerobe would just be
Only aerobic style. So that's what allows a microbe to be facultative, is if it is able to run both styles of electron transport. And then the decision on which one to use comes down to the aerobic makes more ATP. Anaerobic makes less ATP. So if you can do both and you had the choice, which one would you do?
Oh, okay.
Aerobic. It's going to give you more energy for each sugar. If you have the choice, you're going to use aerobic. And if you've got the choice, you make more ATP, and that means that you would grow faster in aerobic conditions than anaerobic. If you've got the choice, if you're a faculty anaerobe. One other thing about this, and you can write it down here. So you circled these and called it final electron acceptor. Now circle, go back. Circle these, this row, water, nitrite, sulfite, and make this say waste products. So we do anaerobic respiration as humans, and our waste product out of that is water and also CO2. So bacterial waste products if they're running anaerobic respiration, the waste products are CO2 back before this. And a lot of times H2S, which is hydrogen sulfide gas. Hydrogen sulfide gas. It stinks. And well, we test microbes in the lab for their metabolic properties. And one of the things that's easy to see or smell or detect is the H2S. So if they're making H2S, that's because they have anaerobic possibility. They're using sulfate as an electron acceptor.
And then you said the other waste product is CO2, correct?
Yeah.
Aerobic.
So, aerobic also makes CO2 as a waste product. Yeah. So, before I talk about fermentation, I'll look for something here. Final things to write about aerobic and anaerobic electron transport chains, which is how many ATP they make. This info is about electron transport chains. In bacteria, where is the electron transport chain happening?
Cytoplasm.
Kind of.
So your liquid or
It's in their cytoplasmic membrane. So in people, it'd be in the mitochondrial inner membrane.
Mhm.
But for bacteria, they have to use the membrane that they've got. So for bacteria, make a big old bacteria. For bacteria, it's happening in the cytoplasmic membrane. This is different from what you'll learn in A&P because that's humans and we have mitochondria. And for us, it's in the inner mitochondrial membrane. But for bacteria, no matter what style of electron transport chain they're running, it's happening in the cytoplasmic membrane. If in an aerobic process, and the number of ATP that you make during the process is up to 34.
That's for aerobic.
Aerobic in the electron transport chain. So when we add in the total ATP, when we add in glycolysis and Krebs cycle stuff, aerobic respiration in bacteria can make up to 38 ATP. And this is a different number from humans. And I'm just sorry about that. It's annoying, but a really efficient aerobic can make up to 38 ATP per glucose. And for the anaerobic ones, they're going to use some other electron acceptor that's not oxygen. And they can make total up to 36 ATP per glucose, which makes this number 32. Some of them may make 8, and some of them may make 24, and some of them may make 32. But overall, aerobic is better. aerobic is more efficient at making ATP. So if you're facultative anaerobic and you can do both, you would choose aerobic.
The anaerobic makes 28 ATP, right? Before you add all of it in, and then 32 altogether.
No, the anaerobic makes up to 32 just in the electron transport chain, and up to 36 when you count the
Okay.
previous.
So it's 2 less.
Yeah, it's 2 less overall. So what I want you to know, oh, go ahead.
Okay. So are we getting, because how you said how some anaerobics make like 8 to like 16, 24, and 32. Do we just need to know the final number of 32, or do we need to know the specific anaerobes and which ones make the specific amounts of ATP?
Just the up to amount?
Just the up.
Yeah. I'm not actually sure what the lower number is. I don't keep up with that level of research, but your book is saying it yields a maximum of, and then they just say anaerobic respiration yields less. But it's up to 32 ATP per glucose. Good question. So I'm not going to try to do fermentation today, but what I want you to know out of this section is what are the steps, where do they happen, like where is it happening in a bacterial cell? This is microbe, so bacterial cell. How many ATP and NADH, FADH2 are made in each step? Okay. What's the difference between aerobic and anaerobic?
And then Friday, we'll talk about fermentation and then go to the next slides.
Monday. This is Friday. Yeah, Monday I will talk about fermentation because now this is the thing I was looking for. So annoying. Okay, this is the chart I was looking for. That is a really good summary chart of these 3 styles. So fermentation we'll talk about Monday, and I'll finish these slides Monday. Okay. And you should also use the ones after this, whatever that is.
I think it's the virus one.
Yay, viruses!
Okay.
This is probably the toughest. No, I think we've got. I think that's Monday, and then I think viruses. But whatever is the next slide set. And if you're not sure, ask me. Ask me. Thanks, and have a great weekend, and please stay healthy and distant.
Where do we find the recording? Do you have to
I
upload
put the
them?
recordings in the page that they go
The
with.
page of the day?
Yeah,
Okay.
which on this day, it would end up in the 916 page because
This was
we're
supposed to be.
shifting.
Yeah.
Yeah.
And then you'll also redo the